Hey Gators, I'm Mr. Ross. I'm a lover of all things science, and I'm a biology teacher here at Roseville High School. And I'm also Dex's and Tyson's dad. You might recognize me as that guy. So here's my classroom. You want to see this? So I teach mostly freshman biology and AP biology here at Roseville High School. That's my setup here. That's where I spend my days. And I heard your class was learning about structure and function of living things. And I thought it'd be really interesting if we did a little DNA lab together. Funny story. My doctor last week told me my DNA was backwards. And I was like, and? So, as a certified biology nerd, one thing that always amazes me is how all living things that have ever been on this planet, whether it's a blade of grass or a mosquito, even a little bacteria cell, or maybe some platies in my fish tank over here, if you can see those guys, or George here. He's been quiet for a while. All these things, even you, even this little fetal pig in a bag. It's sad. I dissect that guy. Or maybe even this sheep brain. Ooh. Or maybe these cow eyeballs. You can dissect some of those. Ooh, I have one more fun thing. I got a pig heart. Fun. All these things that were once living, some that are still are, started as one cell. You should think about, how did that one cell become millions of cells in these fancy tissues doing sophisticated things? How did they know how to do that? Well, the answer is DNA. So DNA's the instructions to your cells, teaching them how to do their job, how to make you grow, how to make you respond to things. It's like the blueprint of a house, but for your cells. All right. So with these cells, or with this DNA, I should say, it's microscopic. It is really, really small. And it's actually pretty basic. So all these living things that have DNA, all this DNA has the same structure. It's really, really simple, actually. It's a bunch of A's and T's and C's and G's. There's 4 different types of what we call nucleotides. And the only difference between you and your cat is that your DNA might say AAATG, but your cat's DNA might go AACCT. That's the only difference. It's kind of crazy, huh? All right. So what I want you thinking about, if this is microscopic, let's say I'm holding this string. Let's say you were half a mile away. Do you think you could see this string from half a mile away? Probably not, right? But what if I took millions of these strings and kind of smashed them together into a big blob? Do you think I could eventually get it big enough where you could see it from half a mile away? Maybe. It's possible, right? That's the same concept we're going to try to do in this lab. All right, we're going to do a lab where we try to pull out all the DNA from a strawberry and get all that DNA to come together into one pure blob of DNA. And hopefully it'll be big enough you can actually see this pure DNA with your naked eye without even a microscope. That's the idea. All right, so I dropped off a bunch of supplies with your teacher. I'm going to kind of show you step by step how to do it. There's a little lab sheet that goes with it if you want to use that too. All right, the first thing you should do is actually a little pre-lab with a couple questions. All right, about where is DNA found? And then also, what do you think it's going to look like if this actually works? All right, so keep in mind, we're going to use a strawberry. You really could use a lot of different fruits. Strawberries are cool though because they're octoploid. That means they have 8 copies of every chromosome. Whereas most animals, especially, only have 2. You got 1 copy from mom, 1 copy from dad, right? Strawberries have 8, which means they have a ton of DNA, which means hopefully we're going to get a bunch of stuff to see, right? Sometimes I do this lab with bananas too. That works well too. All right. So we're going to get your lab supplies in a place where you can access them with your lab teams. And I'm going to walk you through this process step by step. All right. Okay. So you have the instructions on your lab sheet. So follow them carefully. You can pause this video if you need to and kind of go at your own pace. All right. But this is the idea. I'm going to show you what's happening here. All right. You have a strawberry. So you can cut off the stem. No, you don't want the stem. You're going to take your strawberry. You're going to put it in a sandwich bag. Just going to drop it in there. Seal it up. You don't want a bunch of air in there because then it could pop. And just mush it. Try to mush it with your fingers as much as you can. You're basically trying to rip apart those plant cells because plant cells have cell walls that are really tough. So you're trying to break those cell walls, allowing the rest of the cell stuff just to kind of get exposed a little bit. All right. You're going to mush it around for about a minute. Let's try to get that strawberry into a kind of a gooey pulp. All right. You're trying to break open all of those cell contents and really it's really mostly about the cell wall, getting it broken apart. All right. So once you do that for a minute, now you're going to add 20 milliliters. So you guys will have these little cups, and there's a little metric on there. You'll see the bottom line with a number is 30 milliliters, but there's 2 lines underneath that. So you can do the math. If it takes you 3 lines to get up to 30 milliliters, which line would the 20 be on? You can figure that out. All right, but you should have 20 milliliters of what's called extraction buffer. All right, this extraction buffer is 3 things. It's got water. It's got dish soap, just a couple drops of any kind of dish soap that you might use at home. All right, the dish soap is there because it makes things slippery. And DNA is in the nucleus of the cell. It's kind of like the core, the pit of the cell almost. And it has its own special membrane that it's enclosed inside of. And this membrane is made out of lipids. It's like a lipid layer. And the soap is so slippery that the lipids kind of fall apart. They can't hold on to each other as well. So they fall apart, and guess what? DNA gets to sneak out. So now the DNA will get out that way. All right. We also have in this extraction buffer salt. I put a little salt in there because it makes it more gritty and abrasive. So that helps just kind of give extra friction and just help rip everything in the cell apart. Because we just want everything to fall apart and everything get exposed into this big like soup of cell stuff. All the chloroplasts and lysosomes, like ribosomes, all this stuff is just exposed all over the place. All right, so I'm going to put this 20 milliliters of extraction buffer into the baggie. Pour it in. Again, seal it. You don't want too much air in there because then it might pop and make a big mess. All right, and now you're just going to massage it for about 2 minutes. Use your hands, squish it. I wouldn't pound it because then it's going to pop, but squish it with your fingers as much as you can for about 2 minutes. You're just making a little strawberry smoothie. Even smells kind of good. Thought of that soap might take a little drink. All right, so you're really just trying to pulverize it. You're just trying to rip apart all those cell parts, make the membranes fall apart with all that slipperiness. All right, so you're going to do that for about 2 minutes. You just want to get all the solid chunks out of it, really. All right. Hopefully that salt's helping too. All right, once you get all those chunks out, now you're going to build your filtration device. All right, you have a test tube rack. This is just a little device that holds the test tube so they don't fall over. This is a test tube, this glass. And this is a funnel. So you're going to put the funnel in the test tube like that, because we want to separate out all these solids and liquids. The DNA is going to be in the liquid part, and a lot of the excess cell parts are going to be in the solids. All right, so we're going to separate it out by filtering it. So you're going to use a coffee filter, just like you make coffee with at home sometimes. A little coffee filter paper. All right, it's important that this gets wet. So you got to find some water, like I have a sink here. I just get it a little bit moist, All right, and I very carefully just kind of push it down into the funnel so it makes a little bowl. All right, so now I can pour all the strawberry stuff into there. The solids will get stuck on the towel, on the cloth, but the liquids can seep through into the test tube. That's the whole idea. All right, so we're just going to let that sit there. I'm going to take the baggie. Hopefully all the solid chunks are out now. I'm just going to open up the baggie, and very carefully, you just pour this into your little bowl you made, your little filtration device. Kind of go slowly so it doesn't spill over. Hopefully you'll see it start to drip into the test tube. Mine's actually going kind of fast. All right, so I can see it dripping into there. It's pretty pink color. All right, sometimes you'll need to give it a few minutes, maybe up to 5 minutes, but really you just need to go until it's about 2 inches deep. All right, so let it sit for a couple minutes until it's about 2 inches deep. Mine actually went super, super fast. Okay, when it's done, you're just going to pull off your little coffee filter, put it in the garbage carefully so it doesn't spill. You're going to save your little funnel for later. And all you need is that liquid that's in the test tube. Because now you're going to take this 90% isopropyl alcohol. This is what you might put on a cut, right? Cleans the wounds. That's what this is. All right, it's ethanol. So you're going to take a little bit of this. You might have an adult help and make sure this is super ice cold. This has to be fresh off the ice or out of the freezer. And you're going to use this little graduated cylinder to measure 10 milliliters, which almost goes to the top. 10 milliliters of the alcohol. And you're going to, let me take my test tube here. You're going to very carefully pour that rubbing alcohol into your test tube with all your strawberry goodness. You can very clearly see there's 2 layers, right? It looks like they don't even mix. That's weird. Okay, what's happening is that the DNA hates alcohol. It hates the rubbing alcohol. It's just like, get away from me, bruh. Just lose it. All right? But the DNA has nowhere else to run except to other DNA molecules. So that's what it does. The DNA just finds itself. All right? So you're going to take a little stick, a little coffee stir, and very gently just put it down into the test tube and gently stir it right where the 2 layers meet. And if you look super carefully, you might see something is sticking to the stick. You have to look really carefully. It kind of just looks like little bubbles or snot almost. And if you even pull it up, you might even see it. It might even come out. Ooh. Might even come out a little bit. See that? Maybe. I think it looks like snot. Pretty gross. Maybe that's what's been in your nose all these days. All right. So you can just kind of twirl it around a little bit. Take turns pulling up the stick very gently and just look for that little clear substance. See right there? It looks like snot. That's nasty. What? That's cool. See if I can pull it out all the way. Oh, look at that. Oh, nasty. So gross. That's pure DNA, my friends. That is 100% pure strawberry DNA. Okay, so now on your lab sheet, you guys have some analysis questions. I want you thinking about, where is DNA? And is it in the food you eat? Think about that really carefully. Because if you're eating good foods, you're eating plants and animals mostly, right? Those plants and animals were living things. Every single cell of every living thing has DNA. So when you're eating DNA every bite. Might not realize that. It's kind of weird, right? Does that mean you eat a steak and now you have the cow's DNA, so you're like, moo, moo, I'm a cow now? No, probably not. All right, your body's going to take those nucleotides of DNA and break it apart into smaller chunks, and then your body can reuse it again for later stuff. You can use it to rebuild new molecules, new DNA. All right, so with this lab equipment, clean it up in the way that your teacher is asking for. Most of the supplies you can keep and reuse. All right, I hope you guys had fun. I hope you learned a little bit about DNA. There's all kinds of fun stuff you can do with this. And until next time, see you later, Gators.